So we'll be starting now, and I talked to some students. Is it the volume too much or good? Okay, so I talked to some students last class after it ended, and then it seemed like there was some confusion about the different types of LTP, so I'll go over that again slowly. And please feel free to ask questions in any moment. And then after that, we will delve into the actual biophysical and biochemical mechanisms that control LTP and LTD. Okay, so the first concept that we talked about that seemed to cause some confusion was this one here, which is the concept of Hebbian plasticity. So this is not something that's happening in a specific circuit or a particular experiment, but this is like a theoretical framework that makes us easy to understand many different types of phenomena. And basically what they're showing here is that if you have this output cell at the bottom, and let's say before the learning related experience happening, the synapse from cell B is weak, so when cell B fires an action potential, the output cell does not fire an action potential. Okay, so because the synapse is weak and it's not enough to activate the cell, the output cell. Okay, so in the learning related experience, you have this kind of conjoined activation of both inputs, and then that activation is strong enough to activate the output cell. So the output cell fires an action potential. And at this moment, the learning related experience, the output cell has this experience that when cell B fired, I also fired. So cell B is an important input that I should pay attention to. So then it strengthens the input from cell B. And then after the learning related experience, when cell B fires, the output cell also fires. So that synapse has now been strengthened. And this happens in essentially all sorts of associative learnings when, say, you associate this tone with some food coming or this light with an electric shock and things of that nature. And this is not something, this drawing is not about the hippocampus or the amygdala or some region. It's just kind of like a theoretical circuit that could happen in many different brain regions, including the hippocampus and amygdala. Okay, so this is the Hebbian associative plasticity kind of theoretical framework. And then there's this other thing we talked about, which was the here, which is the is it here or no? Sorry, here, which is the specific protocol of Hebbian long-term potentiation in synapses. So this is not a theoretical framework. This here refers to a specific protocol in a real slice in a real hippocampus that was done under specific parameters. And they kind of use that theoretical framework from Donald Hebb to infer what could be a stimulation protocol that would produce plasticity under those circumstances that we saw in the diagram before. So here, what they did is they kind of activated the pre- and postsynaptic sites simultaneously. So they activate this yellow presynaptic cell and also the postsynaptic green cell somewhat simultaneously. And then the green cell has this experience that, oh, when the yellow cell was active, I also got depolarized, so the yellow cell's input is very important, so I'll strengthen the yellow cell's input. And that's what the cell does in all of these trials here. So from 0 to 20, you're only doing a test stimulus on the yellow cell without stimulating the green cell also. And then here from 20 to 25, they are pairing the stimulation of both cells. And then gradually you see that the response size of the same stimulation of the yellow cell increases because the green cell is increasing the synaptic strength from yellow to green. And then one important difference to note here compared to other LTP protocols is that here they just did very few stimulation trials that were paired, like as many as you have those purple points increasing, just something like 10 or 12, something of that order. And there wasn't any strange 100 hertz stimulation, which is a very strange thing that's not physiological. So here, if you just pair the stimulation of the pre and postsynaptic sites a few times, then the cell kind of learns that this is an input that predicts my output, so it increases the strength of that input. So this is the specific Hebbian LTP protocol. And as you see, it's kind of theoretically related to the concept from that diagram that Hebb drew, which is that when cells fire together, they increase their synaptic efficacy. And this is in contrast to the other types of LTP, which were shown like here. So in this LTP, the difference is that they're stimulating only the input cell, and they're just recording in the output cell. So if you stimulate only the input, then you have to stimulate the input very strongly so that the output cell has some depolarization that's synchronous with the input. And that's why they need this very strange 100 hertz stimulation here. It's a very strong kind of strange electrical stimulation that doesn't really exist. And similarly here in the LTD, also only the input was stimulated at 1 hertz, which is a low frequency stimulation. So that's the difference between this kind of normal LTP protocol and the Hebbian. So in the Hebbian, you are stimulating the pre and postsynaptic sites, and you don't need many trials, and the trials don't have any strange, enormous stimulation. And here there's only kind of one learning experience, but it's a very intense 100 hertz, and you're only stimulating the input and not the output cell. Okay, so any questions about how those two protocols are different, or? Yeah.
Is this one similar to the experiment 3 slides later?
Sorry, can you say it louder?
3 slides later from
Three
this
slides.
slide. Is this slide similar to this one? Yeah.
Yes. Yeah, but the LTP that they did in the purple cell is the same as in the previous one they were talking about. And the difference here is that they are doing it to one input and not the other. But what they are doing here to the purple cell is the same as the previous one we're talking about. Yeah. Okay, any other question?
One question about the theoretical framework.
About the Hebbian?
Yeah, the heavy.
Okay.
So for the, I guess the last part of the diagram, the learning that occurs, and that strengthens the synaptic input for B, is that like is that from like repeated or increased fire from synapse B, or is that from?
Is it to increase what?
Like, is it from increased fire of the
Firing?
Neuron B, or is it because of the simultaneous input from A and?
Wait, are you talking about the diagram or the Hebbian LTP?
The theoretical framework.
Oh, okay. So this one here.
Yes.
Wait, and then your question was?
Yeah. So you said like learnings occurred on the last one, right?
Yeah.
So now the output cell will actually fire when it's stimulated by neuron B.
Yes.
But I was wondering if that's because of the simultaneous input that happened before.
Yes. It's because of what happened in the middle time point.
So then, would that also mean that like synapse A would also have the same like impact, or is it specifically like B as opposed to A?
Yeah, here they're not showing what happened to A, but if A was also weak before and then with the pair stimulation, it was enough to activate the output cell, then the A would also be presumably strengthened. But it could also be that the A is already very strong and capable of driving it. But that's not written in the diagram. But if it was symmetrical to B, let's say, then yes.
Okay.
Yeah, so all of these results can really be inferred just by one general rule, which is that the cell is trying to increase the synaptic strength of inputs that predict its own firing, both on the Hebbian LTP and the other type. Yeah.
So when you only stimulate the presynaptic axons, what if the postsynaptic depolarize anyways?
If you only stimulate the presynaptic axon, will the postsynaptic cell depolarize?
Yeah.
It will if it's sufficiently strong, the depolarization of the input.
So if it depolarizes, how is it different from heavy and LTP? Because you're also depolarizing.
Yeah, so that's the thing. So here you're not depolarizing the output cell directly by putting a current in the output cell. It's being depolarized by the input firing a lot and releasing a lot of glutamate. And in the other case, which is the heavy and LTP, you are injecting physically positive current inside the postsynaptic cell. So that's the difference. In this case, the cell in CA1 is only being recorded from. No current is being injected in. And in the heavy and LTP, you have current being injected in and also being recorded from the output cell. Okay, so any other questions?
In this slide, we're looking at the LTP of Schaefer collateral synapses. I was wondering what's the difference, and because a conclusion from that study was that the second pathway is not automatically strengthened.
Sorry, so let's just go to the first correct slide. Is this slide?
That one.
Yes.
It was that pathway 2 isn't automatically strengthened just because pathway 1 was stimulated. So in this, were they stimulated at separate times? Is that the distinguishing factor between?
They did nothing to pathway 2 here.
Okay.
Essentially, they're just doing the test pulse to measure its strength periodically, but they never really stimulated pathway 2 in a way to cause LTP.
Okay, so that electrode going into stimulus 2 is not really a stimulus, or?
Yeah, it's more, well, there's kind of two types of things this stimulus electrode does. One is it can just make a very small test pulse to see what type of response do we get in the cell, which is all they did in the orange. And then the size of the response doesn't change. And then the purple, they did that from -15 to 0. And then at 0, they did that enormous 100 hertz stimulation. And then after that, they resumed the test pulses. And then the test pulses you see are the same small test pulse now gets a bigger response. So it's not exactly they did nothing on stimulus 2. They just did the test pulse, but not the learning experience. Okay, so any other question? Okay, so, and then this slide is just showing that in LTD you also have the same effect where if you cause the LTD protocol on one input, the other input does not automatically get LTD, but it stays where it was. So both the LTP and LTD are specific to the pathway in which the stimulation happened. And of course this makes sense, right? Because learning wouldn't really occur in any powerful, useful way if all the inputs from to this cell are strengthened and weakened simultaneously. It only would make sense if, oh, well, I'll strengthen this little synapse and I'll weaken this other one and so forth. So it has to be very specific for each synapse. Okay, so now let's go to the, also I'll just go over this second example once again because that also seemed a little complicated. So basically what's shown here is that if you strongly activate pathway 1 only, then pathway 1 only gets strengthened as we saw before, so that makes sense. And now here is an additional little twist, which is if you strongly activate pathway 1 but weakly activate pathway 2, then from the point of view of the cell, both pathway 1 and pathway 2 are now correlated with my own activation. So it strengthens both pathway 1 and pathway 2. So the pathway 2 is not really doing most of the heavy lifting that's being done by pathway 1, but pathway 2 is still a predictor of the cell's output. so pathway 2 also gets strengthened, and that's what's being shown here. And this is called the associativity or cooperativity feature of LTP. Okay, so any questions about this part here? Yeah.
Are they strengthened to the same extent?
Yeah, so it's difficult to say they're strengthened to the same amount because these measures are always relative to their baseline, right? And it's difficult to have some absolute value for that because they depend on some experimental factors that are not what you want to be measuring. So for example, it could be that the electrode is just more near more cells in pathway 1, so it stimulates like 50 axons, and here the electrode stimulates like 7. So that it couldn't be that, but and you don't know exactly how many axons are stimulating, what's the distance from the electrode to all the cells. So there's a lot of little non-controllable factors that make it difficult to kind of compare the absolute value of how much it increased in strength. Also, if the pathway 1 is already super strong, then it has also less room to increase. So there's also that. Okay, so any more questions about this slide here? Okay, so now we can go into the actual biochemistry of how these processes work. So the first result here is on the black circle. The filled black circle is the control condition. So they made LTD happen, depression, and then depression happened. And then the white circles are if they did long-term depression in the presence of AP5, which is a drug that is an antagonist of the glutamate NMDA receptor. So the glutamate receptor has kind of two subtypes that are very important in this class. One is the AMPA subtype and the other is the NMDA subtype. And AP5 is a drug that is an antagonist of the NMDA receptor. So what you see is that the LTD doesn't really last long if NMDA glutamate currents are blocked. So this was the first major discovery of this pathway, which is that it requires activity of the glutamate NMDA receptor to happen. And then the second discovery is here, which is that it also requires the activity of a phosphatase to happen. So phosphorylase is an enzyme that puts phosphate groups on target proteins, and phosphatases remove phosphates from target proteins. And okadaic acid is an inhibitor of a phosphatase, and in the absence of that phosphatase working properly, you don't get LTD. So this shows two very important biochemical results, which is that you need both the NMDA receptor activity and phosphatase activity for long-term depression to happen. And through kind of experiments of this sort, people have figured out conditions that slightly increase or decrease LTP and LTD and what chemicals and signaling pathways are necessary for these processes. And here again, we shouldn't think of this just as some strange thing that's happening in a brain slice in a petri dish, but rather these same pathways are being activated in us every day when we are experiencing things and acquiring new memories. And if we didn't have these pathways working, there would be like profound impairments to how our life is. So for example, if we just didn't have one of these proteins, like if we didn't have NMDA receptors, then we wouldn't have normal functioning hippocampal memory. And your life would be in all sorts of disarray if you didn't have this one protein in the hippocampus working properly. And so now we're going to see how this NMDA receptor works. So this is probably something that you might have been told in some previous class. So has anyone ever learned about this receptor before? Oh, cool. Okay. Does anyone remember well how it works? Or okay, so let's give a brief overview again. So this is a ionotropic glutamate receptor. So ionotropic meaning that when the neurotransmitter binds, the receptor itself is a channel, so it opens and then ions go through, so it's an ionotropic receptor. But somewhat complicated ionotropic receptor because it requires two conditions to be activated. The first is glutamate has to bind the receptor because it's a glutamate receptor. And the second is that the cell has to be depolarized. And that's because if you see there's a little channel because it's an ionotropic receptor. So that central pore is through where the ions will go. But there's sort of a clog on the pore. And the clog is a magnesium ion, which is this ellipsoid thing in the middle. And the magnesium ion sits in the pore like a clog inside of some plumbing and impedes the flow of the ions. And the magnesium is positive because magnesium 2+. So when the cell is depolarized, so there's a lot of positivity inside the cell, that positivity repels the magnesium clog and the magnesium goes out, and then the pore is free to conduct the ions. So for this receptor to be activated, you need a coincidence of two things to happen. Glutamate has to be released and bind the receptor, and the cell has to be depolarized. Okay, so does anyone have a question about how this receptor works?
What is the function of glycine?
The glycine is like a cofactor that also has to be bound there. So it has to be, and it's a also normal fast neurotransmitter that's present in many synapses. So there's kind of a third factor, which is this glycine binding there as well. But for the purpose of associative learning, the two more important factors are the depolarization and glutamate presence. And we should assume kind of the glycine already is there in all the situations we're talking about. Okay, so why do we need this complication of having a magnesium blocking the pore? Because it seems easier just to not have a clog inside the ion channel. And that's because this allows this extra complexity, which is now the cell will only be active when there's this coincidence of glutamate release and depolarization. And then we'll see in the next slides how that is useful for learning. So through those patch clamping kinds of results in brain slices, it was found that both LTP and LTD require activity of this NMDA receptor. And that sounds very strange because LTP is increasing the strength of the synapse and LTD is decreasing it. So it's strange that they both depend on the same channel. But the reason is kind of shown here. Okay, so when there is long-term potentiation, you get a lot of glutamate release from the input, right? Because you have that 100 hertz enormous stimulation. And then you have a lot of glutamate release. So then the NMDA receptor conducts a lot of calcium inside the cell, which is many red dots. And then if you have a lot of calcium influx through this NMDA receptor, it activates this pathway through calmodulin kinase 2, which creates some effects that lead to long-term potentiation, such as the insertion of new glutamate AMPA receptors. So if you have more AMPA receptors in the synapse, that synapse will now respond more strongly to glutamate being released onto it. So this is one common way of strengthening synapses. Okay, and then on the long-term depression end of things, we have very low 1 hertz frequency stimulation, and then you get very little calcium accumulating through the NMDA receptor influx. So we have fewer red calcium dots here. And then if you have very few calcium ions that come in, it activates a different pathway and activates this protein called calcineurin. And then the calcineurin does the opposite. So the calcineurin takes back AMPA receptors from the synapse and pulls them back into vesicles, which weakens the strength of this synapse. And the calcineurin, which acts in depression, also dephosphorylates the AMPA receptor, and the dephosphorylating also decreases its kind of efficacy. And that's why that phosphatase inhibitor from the other slide was important for long-term depressions, because phosphatases are taking out phosphate groups from the AMPA receptor, which decrease its excitation power. And the CAM kinase is both putting in more AMPA receptors and also phosphorylating AMPA receptors, both of which increase the synaptic strength. But let's see in more detail how each of those things work. So here we have a diagram summarizing how long-term potentiation works. Okay, so you get release of glutamate. Then if the stimulation is strong enough to depolarize the dendrite here, then the magnesium ion will pop out of the NMDA receptor, and you'll get calcium and sodium going through the NMDA receptor, and the calcium is the most important thing that goes through the NMDA receptor. So you get a lot of calcium going in because it's a very strong depolarization of the input. It activates this calcium calmodulin protein, and then the calcium calmodulin will activate a pathway that puts that P phosphate group on AMPA receptors. So if you phosphorylate the AMPA receptors, the Ps, then the AMPA receptor will conduct more ions per glutamate bound to it. So it becomes kind of a better AMPA receptor that conducts more positive charges. So phosphorylates the AMPA receptor, and it also activates this CAM kinase 2 protein, which leads to the insertion of new AMPA receptors. And the third thing that happens is that you activate these other pathways that are dependent on cyclic AMP and MAP kinase, and then they go into the nucleus of the cell and activate the transcription of target genes. And then those genes that are transcribed produce proteins that are necessary to make new components for new synapses. And this is something pretty cool because, usually we tend to view these two things as very separate. Like you think that, cells have some electrical activity in their fire action potentials, and that involves this world of channels and sodium ions and so forth, and that's one type of event that happens. And then we tend to think there's this other different type of thing, there's genes in the nucleus, and then it produces mRNA and proteins, and we tend to think that those two things are separated, but in reality those are working in the function of the same cell, so they must be integrated somehow. And this was one of the first studies that found out how those things might be integrated, which is that when you get plasticity, you also get the kind of communication from electrical activity of the dendrite into transcription of genes in the nucleus, which produce new proteins that are necessary to build more synapses. Okay, so that's what's happening in the potentiation end of things. And here we have a before and after picture of what happens before and after the LTP. So many things can happen. So one is that first the response size gets bigger, but also, see, there are changes that can happen both pre and post synaptically. So the presynaptic change in this case after the potentiation is that you have more vesicles. So when this purple cell now fires an active potential, like more vesicles will fuse, so it will release more glutamate. And then on the postsynaptic end of things, we have more AMPA receptors. So we have both more glutamate being released and more AMPA receptors. And in addition, those AMPA receptors can be phosphorylated, which further increases their conductive abilities. So all of those three things taken together are some of the major things that are happening during the long-term potentiation. Okay, so any questions about this part here? Yeah.
So presumably if LTP is activated by coincidence of these things, how does the presynaptic neuron know coincidence happens in the postsynaptic?
Oh, yes, very good question. Because, so did everyone hear what he asked? And so he asked, if LTP happens because the two cells need to be coincidentally active, then the green cell kind of knows it was coincidentally active because it can sense the glutamate from the presynaptic cell, and it also is in control of its own action potential, so it has this information. But the purple cell doesn't really know that the green cell fired. in principle. So how does it know to strengthen this synapse? And the way it's doing that is through a retrograde signal that goes from the green cell to the purple cell. And that retrograde signal, because usually the signals are going from pre to postsynaptic, but there are also some retrograde neurotransmitters that go in the opposite direction, and that neurotransmitter in this case turns out to be a gas, which is nitric oxide. So nitric oxide is released from the green cell and goes to the purple, and then the purple is like, oh, we are synchronously active, so I should strengthen this synapse. So now let's look a little more in detail to the actual proteins that create this strengthening of synapse. So the most important protein after the NMDA receptor that we'll talk about is CAM kinase 2. So it looks kind of like a little hair clip, like it bends like this. And when it's in this conformation, it's inactive, and it opens like this, it becomes active. And it opens when you get this calcium influx to the NMDA receptor. Then the calcium activates calcium calmodulin. So calmodulin binds the calcium, and then the calcium calmodulin complex binds the inhibitory blue domain of CAM kinase 2, opening it. And then after it's opened, it can autophosphorylate itself. So it puts a phosphate group here on itself. And after it becomes autophosphorylated, it can now stay active even in the absence of the calcium calmodulin. So first it's inactive, then you get calcium influx, then the calcium binds calcium calmodulin, then the calcium calmodulin opens the paperclip, and now when it's open, it can autophosphorylate itself, and now when it's autophosphorylated, it can stay open even if the calcium calmodulin goes away. Yeah.
I have a question. So both LTP and LTD depend on influx of calcium.
Mm-hmm.
So wouldn't LTD also activate calmodulin too, or is it like a?
No, it doesn't, because it has a kind of lower calcium binding affinity. So it only would, calmodulin only bind calcium if there's a lot of calcium. Because if the calcium calmodulin bound calcium, if there's like one calcium around, then that's what would happen. But this works because the different pathways have different binding affinities for the calcium. Yeah, but that's a good question, because this was a big mystery when this came out, because people found out first that, oh, calcium is required for LTP. So then everyone was excited about that. Then they discovered calcium is required for depression too. Then everyone was very confused. And then it turned out because these different pathways have different binding affinities for calcium, so they are sensitive to either low or high calcium influx. So the CAM kinase 2, which is this hair clippy shaped thing, it binds other CAM kinase 2s and forms this 6-member hexameric ring sort of structure. And each kind of unit can phosphorylate the other one. And when they're all phosphorylated like this, they can act and do their thing of putting more AMPA receptors and phosphorylating the AMPA receptors and activating those pathways that go into the nucleus. But they don't just stay activated forever, because then you'd just be strengthening synapses forever, which wouldn't make sense. So they are constantly being inhibited by phosphatases, which are these square blue proteins there. So the square blue proteins are phosphatases, which remove the phosphates, inactivating each subunit of the CAM kinase 2 complex. So there's always some kind of push-pull tug of war between should we strengthen this synapse or weaken this synapse. And sometimes one of them wins, sometimes the other one wins. So we shouldn't think of these as either LTP happened or LTD happened, but the more common thing in neuroscience in general is that there are opposing forces that are active at the same time always, and then in some moments one of the forces wins, and some other moment the other force wins. So there's always this balance between phosphorylation trying to happen versus dephosphorylation through the phosphatase trying to happen. Okay, so any question about this slide here? So So GluR1 is a subunit of the AMPA receptor that is being phosphorylated. So when the AMPA receptor is phosphorylated, it's being phosphorylated here in this subunit called GluR1. And phosphorylating this is what strengthens the ionic conduction potential of the AMPA receptor to increase the synaptic strength. So this is a kind of diagram showing how the phosphorylation there is happening in LTP and LTD. So first we see that the CAM kinase 2's activity increases with a high frequency stimulation. So that's CAM kinase 2 is again that protein that is the hair clip that organizes in a 6-member ring. So its activity increases after the high frequency stimulation in graph A. Then in graph B, they're plotting the amount of phosphorylation in CAM kinase 2. So remember the CAM kinase 2 here becomes phosphorylated, and that's what maintains its activity. So after LTP happens, it becomes more phosphorylated. That's shown in B. And then in C, you see that if you depotentiate, so do LTD after the LTP, then you get a decrease in the amount of phosphorylation. And on D, they're plotting the phosphorylation level of the AMPA receptor. So GluR1 is a component of the AMPA receptor. And the phosphorylation of the AMPA receptor increases from baseline in blue to LTP. And it decreases from LTP to depression. So this is just an actual measurement of this diagram showing that we get kind of more phosphorylation of the AMPA receptor during LTP, and what precedes that is more autophosphorylation of CAM kinase 2, because the CAM kinase 2 pathway has to first phosphorylate itself, become activated, and then that activates the AMPA receptor. So those are two consequences of the strengthening. Okay, so any question about this graph here? And here we have kind of a model that ties everything together. So in both for LTP and LTD, you get calcium influx with an NMDA receptor. And then if you have a lot of calcium influx, it activates this CAM kinase 2 pathway. It leads to potentiation. If you have very little calcium influx, it activates this calcineurin pathway, and that leads to depression. And they roughly are doing opposite things, right? So the potentiation is phosphorylating AMPA receptors and putting new AMPA receptors on the dendrite, and the depression is dephosphorylating AMPA receptors and taking out AMPA receptors from the dendrite. And on panel B, we see how the amount of calcium is related to the frequency of stimulation of the input axons. So if you stimulate the input a lot, you get more and more and more calcium influx into this dendrite. That makes sense. And then the synaptic strength is kind of weakened with very little calcium influx and strengthened with more calcium influx. So this is the diagram that kind of ties everything together. So yeah.
So is like the difference in the role of calcium for LTP and LTD, does it have to do with the calcium affinity like you mentioned earlier, or is it the concentration of calcium that's influenced?
Oh, it's both. Because so first, yeah, with low calcium you get the depression, the high you get the potentiation, but that happens because those pathways have different affinities for binding with calcium, right? Because if the CAM kinase 2 had a very high binding affinity for calcium, then just a little calcium came in, it would bind it, and then it would cause like a little potentiation. But what happens is the calcium kinase pathway has lower affinity for calcium, so it's only getting activated when there's like a lot of calcium in there. So it's both, and they happen because they're differentially sensitive to calcium. Yeah.
I don't want to go too much into it, but it's at high, but it doesn't really explain why LTD wouldn't also activate at high, because presumably LTD would be higher sensitivity, higher binding of calcium, is what I'm guessing. So when there's a lot of calcium, wouldn't that activate both LTP and LTD?
It would, but they're also competing with each other. Right, so then the chemokines would get more phosphorylated and the phosphatases would kind of lose. Okay. And here we have some details showing the mechanism of expression of LTP. So we have a influx of this calcium to the NMDA receptor, and that activates various pathways, which will eventually lead to the production of new genes, which is something we'll see in the next slides. And then here we have that retrograde factor we mentioned, because someone asked, how would the presynaptic cell know which synapse to strengthen? Right? Because the presynaptic cell also wants to strengthen the synapses for which it is more influential. Like, it doesn't want to strengthen the synapses that don't adhere to its influence. But also the presynaptic cell kind of doesn't directly know when the postsynaptic cell was active. So how would it know which cell is listening to my output? So it knows because of some retrograde factor that has to go from the postsynaptic cell to the presynaptic cell. And this happened exactly like as we were discussing in class. Because first it was seen that, the synapses get strengthened when there's coincident activity. And first it was found that the postsynaptic cell did a bunch of things to strengthen the synapse, like putting more AMPA receptors, etc. And that seemed okay. But then it was found that the presynaptic cell also had some modifications after the potentiation. And then that created this issue: how does the presynaptic cell know which synapse to strengthen? And it knows because it was first just theoretically thought that there must be some communication from the postsynaptic cell to the presynaptic cell, because otherwise how would it know? And later it turned out that they found which was this retrograde factor, and that was nitric oxide. And this also sounded strange to me because nitric oxide is a gas. And typically you'd think neurotransmitters are not gas. They're kind of a little molecule in a vesicle, and they're packaged and they're released and bind a receptor. But there's a very enormous amount of types of molecules that can be neurotransmitters, and that includes fatty molecules and also gas. But in this case, it's not a normal neurotransmitter that's packed into vesicles, but rather it's produced in the postsynaptic cell, and then the gas molecules kind of just diffuse out of the dendrite passively and reach the axon terminal of the presynaptic cell. Okay, so any questions about this part here? So now let's look at a little more detail into how the patch clamping setup actually works. So you start with a rat or mouse that had a head and a brain inside the head, as a normal mouse would. Then you first have to extract the brain, and just to orient you, the back side is here on the right and the front part on the left. Then you have to quickly make a slice containing the hippocampus. Then you put the hippocampus inside this kind of petri dish that has very specifically tightly maintained parameters, so the temperature has to be very super tightly controlled, and the amount of acidity and oxygen and carbon dioxide, all of that has to be very correctly controlled, so that the slice that really wants to die, because it's outside the brain, but you can trick it into staying alive for a few hours to do these experiments. And that's what the thing actually looks like. And then you have this little hippocampus with some cells in there. And the way the patch clamping works is you make a very thin glass pipette with a little hole at the end, and then you literally poke the cell with your glass electrode pipette. And that's a photo of a real hippocampal cell being poked by this electrode. So it's a very strange thing. You're kind of communicating with one cell. And after you puncture the cell, the inside of the glass electrode has a ionic solution that becomes continuous with the cytoplasm of the cell that's being patch clamped. So now the inside of the glass electrode and the cytoplasm of the cell are electrically connected, so you can record what's happening in the patched cell. So this is a very strange thing because you can kind of do little things to one cell and then you can see how the cell responds back. So it's almost sort of you're having a conversation with one cell. It's very interactive. Like you do something, the cell responds, then you can put some drugs that change the behavior of the cell does something else. So people have discovered a lot about how the brain works using these patch clamping preparations. Okay, so then after people saw that there was long-term potentiation, they tried to see if the effects were being caused predominantly by presynaptic mechanisms or predominantly by postsynaptic mechanisms. And it turned out that both things kind of happen, and different experiments teased out components from both pre- and postsynaptic strengthening. So here is an experiment where they made a stimulation electrically. So that first little vertical thing that you see going up and down like this, that's just the recording of the electrical stimulus itself. So that's not some biological event that happened. That's just kind of, if you have two electrodes here and put some current here in a bucket of water, you'll record it there. So there's no biology involved. So that's what that first thing is. Then after that, you get this lower response, and that's the thing that was biologically caused by the cell response. So here what they're doing is they're stimulating some afferent inputs with that stimulation electrode, and then they're recording it with the recording electrode that is patch clamping this cell here. So if you put some strong stimulation, you see a big response, and then put some weak stimulation, you reach this interesting point where that stimulation is so weak that it's stimulating only one or some very small number of synapses. And then if you're stimulating at this barely working amount of stimulation that the cell can barely see the current, then you see this interesting regime where sometimes you get a response, you get this downwards current, and sometimes you don't, which is the flat trials. So the flat trials, when you don't see a response, is kind of a failure. Like you activated this input a little, but you didn't find a response in the postsynaptic cell. And then the downwards one are trials in which you didn't fail. So if you have some very weak stimulation, sometimes it fails, sometimes not, and there's some failure rate. Okay, so the straight line is a failure, and the downward line is not failed trial. Right, so if you plot the number of trials in which you got failure versus not failure amounts of current, you get that distribution in panel A. So you get a bunch of responses with zero current, which is a taller little peak, and you get some responses that have like 4 units of current. And that's the relative distribution of the peaks. And then after you do LTP, you get fewer failures. So you see the zero distribution is smaller on B, and the distribution for current is bigger on the 4 units of current. So you get fewer failures after doing LTP. And then that is because after doing LTP, you get increased probability of presynaptic release of the vesicle. And that is something that's happening presynaptically, right? Because the postsynaptic cell can't do anything to change the failure rate. So if the vesicle is being released more often, that's something that's happening on the end of the presynaptic cell. So this is an experiment that suggested that there is some presynaptic component to strengthening the synapse. So any question about this? Yeah?
If the measurement of success is stimulating presynaptic and then recording postsynaptic, how do you distinguish between whether it's because the more vesicles are being released or because now you have more AMPA receptors than phosphorus?
Yes. This is more because it's shifting from 0 to something. So it's more, vesicles are more times a vesicle is being, and this is kind of one vesicle. This is not vesicles. This is like, it's so weak that you get a vesicle release. But if it were what you said, which is the AMPA receptor is making the cell more responsive to the stimulation, etc., then you'd get a rightwards shift. Like, so instead of getting 4 units of current, you'd get 6 units of current. And here you don't get that, you get more of 4. So the 4 doesn't become 5 or 6 or 7. But related to that, under some other conditions, it was, with slightly different stimulation protocols, it was found that exactly that can happen. You can go from before LTP having current sizes from 0 to 10, to after LTP, the current size can go from 0 to 21. So now if you use slightly different conditions, slightly different stimulation patterns, you can get a postsynaptic measurement of the strength. Because this would be caused by what you were suggesting, which is if you have more AMPA receptors in there that are more sensitive to the glutamate, then the same amount of glutamate would cause more current. And so these two measurements together show that there's both pre- and postsynaptic components to LTP. But the postsynaptic component is much more studied than the presynaptic ones.
Quick question about that.
Yep.
So the reason we know that this one's postsynaptic, like the postsynaptic effect, is because was it like the same amount of presynaptic input, like given to the postsynaptic cell?
Because what?
Like, was it like the same amount of stimulation as before, like before LTP coming from the presynaptic cell? Is that how we know that the postsynaptic cell, like something different happened there?
Yeah, it's the same amount of stimulation before and after the LTP in both cases.
Okay.
But that's true also in the other experiment. But here we know it is postsynaptic because the size of the response becomes bigger. And then later it was found, not in this slide, but through other data, that like if you impede the phosphorylation of AMPA receptors, then this effect goes away, for example.
That makes sense.
Yeah, but you guys are making all the correct questions, which most of which are not shown in the slides, but people have done these controlled experiments to figure out what was happening.
Just to clarify, so we talked about like using more AMPA receptors on the postsynaptic neuron if like if there's LTP. So that would be like in postsynaptic evidence.
Yes.
So we're saying that there's both presynaptic.
Yes.
and postsynaptic.
Yes. Yeah, the more AMPA part is the postsynaptic strengthening, and the lower failure rate is a presynaptic strengthening. Okay, and then there's this even more kind of strange thing that happened, which was that LTP experiments showed that there was a thing called silent synapses, which wasn't known before LTP was being studied. And this and the unsilencing of silent synapses turns out to be another method of potentiating connections between cells. Okay, so what is a silent synapse? So here in panel A, they just do a normal thing of you do a big stimulation of the input, and then you see a big response in the output cell, so everyone is happy and nothing strange happened. Then the second panel in A, the one that says low intensity at minus 60 millivolts, there you do the same thing, but just very, very, very low stimulation. And then you don't see a response, and that's not super weird either, that with some very low stimulation you don't see a response. Nothing too strange. Now here it's when something very strange happened. So now they hold the same cell as plus 30 millivolts. Because usually in the first two panels the cell was clamped at minus 60 millivolts, which is somewhat near its resting state. Now they clamp the cell as plus 30 millivolts, so the cell is very depolarized. And then they applied the same low intensity, very small stimulation that caused no response in minus 60. And then you see some responses at plus 30 millivolts. So this was super weird because the response appeared only when the cell was depolarized. This very, yeah.
I just had a question about the experimental design. So
Hmm?
the minus 60 plus 30, that's the postsynaptic
Yes.
part, and the stimulation is happening on the presynaptic.
Yes. Yeah, in patch clamping, those voltages always refer to the voltage of the postsynaptic cell because that's the cell you are doing the actual patch clamping in and putting the glass electrode inside. So there is some synapse here, but it's kind of silent at minus 60 millivolts, and it only appears at plus 30 millivolts. And that response at plus 30 millivolts disappears in the presence of AP5 in the bat, and AP5 is an NMDA receptor antagonist. So this shows that there are some synapses that physically exist and only have NMDA receptors, because this response goes away with NMDA antagonist. And they don't have the normal kind of AMPA receptor response that appeared at minus 60 millivolts. And the reason the NMDA response appears at plus 30 but not minus 60 is because the NMDA response appears only when the cell is depolarized to get the magnesium clog out, and there's glutamate release, right? So in this first panel, there's glutamate release, but cell is not depolarized. Oh, sorry, the second panel, low intensity. Then on this panel, cell is depolarized at plus 30, and there's glutamate release from the weak stimulation. So then there's a weak NMDA response that is blocked by a NMDA blocker, which is this AP5. Okay, so, and then they showed that if you find such a strange silent synapse and then do kind of normal LTP, you unsilence the silent synapse after doing LTP. And that's because after doing the LTP, you get the insertion of new AMPA receptors and etc. So now you see an AMPA response that happens at minus 65 millivolts. Yeah, so any questions about this part?
Just a nomenclature. So the
Mhm.
pairing would be like they just paired different inputs, and that's the way that they got LTP, right?
Yes. Yeah, so let's see if this is the details here. Yeah, so it was normal. You pair activation of the input, the output, but the output has to be depolarized, otherwise the NMDA current wouldn't appear to begin with. And then if you return to the minus 60 resting potential, now AMPA receptors have been inserted, and this is now a loud, noisy, unsilenced synapse. And this was very surprising. No one thought there would be such a thing as silent synapses, but this showed that there are some structurally ready synapses there that are ready to go, but they don't work until AMPA receptors are inserted in there. So they're kind of like vacant houses without not connected to the electric grid, but they're there. And this was a very new, exciting development in the plasticity field. Yeah.
Can you please point out where the silent synapses in these
Where the sounds?
The silent synapses.
Yeah, so all of this is the same synapse, but they're just being recorded in different conditions. But the discovery was in this going from the second to the third panel. So in the second panel, they are stimulating very weakly, and then they don't see a response at minus 60 millivolts. So, okay, no response. But then the surprising thing is, that same cell that you're patch clamping that was silent, if you then put in the same weak stimulation for the input, but now while depolarizing the patched cell at plus 30 millivolts, then you see a small little response that is created by NMDA receptors because it's blocked by this AP5, which is a NMDA receptor antagonist. So the synapse so silence really means no AMPA receptors. And it's silent at this normal physiological condition of minus 65 millivolts.
So this finding is obvious biologically, what does it mean, since you need depolarization to trigger LTP and you need LTP to trigger depolarization? And if we talked about that each synapse only strengthens itself, then what would, in like a biological setting, what would activate these silent
Yes. So that's also a good question. So, yeah, you guys have all the correct questions, because when this came up, people were like, why would you even have this? This is very strange. How would this ever get unsilenced in real life, because there's no guy with an electrode making it depolarized? So what happens in normal life, presumably, is that this synapse can get depolarized through some other means, which could be, for example, other neurotransmitters that are not AMP or NMDA, like some neuromodulatory neurotransmitter, or more commonly, depolarization that invades from some adjacent dendritic spine. So you can have a little dendrite like this, and this is a synapse, and this is another dendrite, and okay, and then this one, let's say, is silent, has no AMPA receptors, and then this one has AMPA receptors in it, and it's not silent. Then this one can get depolarized, and some of the depolarization can invade the other one. And this is more likely the more common thing that happens to activate it in real life. Okay. So, and this is a kind of model of how the AMPA receptor trafficking could work to unsilence the silent synapse. So you have some mature synapses that have both NMDA and AMPA receptor. Then you have some silent synapse that has only NMDA receptor. And after unsilencing, you get AMPA receptors inserted either from vesicles or from lateral diffusion. So there's also AMPA receptors that are kind of nearby the dendrite, but not on the active synaptic site. So they're kind of a car in the garage. It's like the car is nearby the road, but it's not being driven. And then when you unsilence the synapse, the AMPA receptors also can migrate from near the postsynaptic site to the postsynaptic site. So they can both kind of migrate to the correct place or also be placed there from a membrane, from a vesicle. And another potential way through which you could get potentiation shown here, it's also possible that perhaps the silent synapse doesn't not have AMPA receptors, but perhaps has very few and very little glutamate is being released into the synapse, which is shown here on the panel A. So one potential model is that when the synapse is silent, the vesicle fuses incompletely in the presynaptic end, and then just like one glutamate molecule comes out. And because the NMDA receptor has higher affinity for glutamate than the AMPA receptor, if there's just a little glutamate that's released, it will all bind essentially to the NMDA receptor, which has higher affinity for glutamate, so it appears silent. And then perhaps after potentiation, you get more complete vesicle fusion, more complete release of the glutamate, and then there's enough glutamate for the AMPA receptor to grab some. So that suggested a potential kind of presynaptic mechanism for the unsilencing. And another possibility is that you have these empty structures sitting there, and then both pre and postsynaptically after the induction happens, you get changes on both ends that recruits these empty empty synaptic sites. Any questions about this part here? Okay, so the last thing we'll talk about is some longer lasting properties of long-term potentiation. So everything that we talked about so far to strengthen the synapse, which was insertion of new AMPA receptors and phosphorylation of new AMPA receptors, are things that could strengthen the synapse for some minutes. but not for many hours. And that was first found in this experiment here, where they showed that some different mechanism was going on when you had very long-lasting long-term potentiation of many hours. Okay, so here they made long-term potentiation, and it looked okay in the control condition, which is the black circles, and lasted hundreds of minutes. But then if you repeat the experiment in the presence of actinomycin D, which is a protein synthesis blocker, then you get the initial rise in LTP, but then the LTP doesn't last as long and decays fast. It only lasts a few minutes and not 3 hours. So it seems that synthesis of proteins is necessary for the late phase of LTP, but not for the initial induction of LTP. Yeah.
Is this like the same protein synthesis blocker in like the fish? That could
Yes, the same thing that they did in the fish. I mean, it's another protein blocker, but yes, protein synthesis blocker. Okay, so, and why do we need this protein synthesis? We need this protein synthesis because after you strengthen the input from one axon to another postsynaptic site, you see the kind of birth of new dendritic spines. So here you have the patch typing recording showing that after you strengthen the synapse, you get this early phase LTP that doesn't require protein synthesis. And then the late phase of LTP, you see a little, in the gray, in the bottom, you see a little dendritic spine blossoming like a flower. So you get a new dendritic spine forming that kind of maintains the LTP into the later phase. And the reason the early phase LTP goes away is because, as I mentioned before, the early phase LTP is created by this phosphorylation, putting new AMPA receptors, etc., and that gives that initial rise in the LTP. But then there's always these phosphatases that are removing the phosphate, putting back the AMPA receptor into the vesicles, etc. And that's why the early phase of the LTP goes away. But the late phase of the LTP is created by kind of micro growth of new structures like these dendritic spines. And we shouldn't think of these dendritic spines as some static thing, but they're rather often very dynamic and they're changing according to learning and habits and things that are happening. So a little dendritic spine might appear here, and then this one might get retracted, and another one appears there, and it's a very kind of dynamic process. Because in the textbooks it just looks like, oh, there's a big dendrite here, there's a big axon terminal there, and they're just there forever like a big building. But they're not like big buildings. They're kind of like little contacts that appear and disappear and strengthen or weaken and so forth. And this was found out when you started imaging these things from a microscope, so you could image the same dendritic spine, the same animal, before and after the animal learned something. So if you can image in cortex, in a part of cortex that, say, is in auditory cortex, then you teach the mouse some kind of auditory task. Then you see some turnover of synapses and increased growth of some dendritic spines, retraction of others, so it's very dynamic. But one thing to remember is that it's dynamic, but you don't get new synapses forming in all sorts of ways, but it's more like a pruning of a local circuit. So for example, let's say you have this brain region A here that projects to this brain region B. Then you could get this little synapse increases and this dendritic spine increases, this one is retracted, etc. Some local pruning of the connection happens and some local growth of little new dendrites can happen. And this is what's mediating a lot of the long-term plasticity. But what doesn't happen is this. Let's say there's this other region C that does not project to B. So after you learn something, you do not get an axon from C that traverses the brain and goes far away to this other region. So all of this neuroplasticity is happening kind of by slight rearrangements of circuits that are already connected in some way. You don't get completely new connections appearing due to learning across brain regions that don't talk to each other before learning. Okay, so we mentioned that there was some kind of gene expression that happened after potentiation happened, and that's shown here. So first, you can induce just early LTP by doing just one kind of stimulation, and then that decays quickly. And if you give 4 stimulations that are big, you can get this longer lasting LTP that lasts hundreds of minutes. So something different is happening in late LTP, and that turned out to be the induction of transcription of genes in the nucleus. So you have this cool communication between electrical activity of the dendrite and ions flowing, that whole world. And that activates CAM kinase 2, as we saw, and then CAM kinase 2 will activate these other pathways like cyclic AMP, which activates protein kinase A, and then that goes to the nucleus and activates some transcription factors that will increase the production of target genes. So for example, one of those genes is brain-derived neural growth factor, which is something that increases the production of new synaptic contacts between neurons. And there's also other things that are just structural components of the synapse, because you have to build a bunch of stuff to make a new dendrite. So that stuff is being produced here when you're activating these target genes in the nucleus. You'll get this communication between electrical activity and genetic transcription. And this is sort of what's happening when you remember anything for some decent amount of time. Okay, so we also have a short and longer lasting memory. Okay, so something that is a very short lasting memory would be, now whenever you log into UCLA, you have to put that annoying code for security purposes, and then you have to remember the code. It's like, oh, this code, and you type in that code, and then you forget the code. So, but there was a memory because you remembered it until you typed it, and then that thing is mediated either by persistent activity of cells firing or fast things like phosphorylation of AMPA receptors. But you don't know that code tomorrow. Like you press hundreds of these codes and then you don't know a single one of them, so you forgot all. But things that you do remember from yesterday or from more longer time points in the past are because those processes or those memories created the production of new proteins, so it required protein synthesis and built kind of new microanatomical connections, and that's why those are more stable than kind of phosphorylating a receptor or making the AMPA go from near the postsynaptic site to the site. So if you remember something from yesterday, it's because there was some protein synthesis in these memory encoding parts of the brain. Okay, so any questions about this part here? Yeah.
For vectors for growth, what is TPA stand for?
So what?
For vectors for growth, what does TPA stand for?
I don't know. I could look it up though. Yeah, so you just email to me and then I can look it up. Yeah, so for our class purpose, we're not really, it's not just 2 gene series. It's like hundreds of things are produced. So everything that we're talking about here is very simplified, and if you zoom in, there's a whole zoo of proteins interacting in all of these steps. So for our purposes, we are more just saying they are genes, they're necessary for the creation and maintenance of new synaptic contacts, and we're not really getting into what each of those are, but I can look that up and then I can tell if you just email me. Yeah.
We were talking about protein synthesis
Mhm.
role in long-term memory participation, I'm sure. And then we saw those experiments where pyrimycin actually blocks the block long-term memory.
Mhm.
Do you happen to know what pyrimycin is actually working on? Is it working on like one of these transcription factors? This diagram, for example.
Yeah, it will be working on, not on the transcription factor, but it will be impeding the translation of any mRNA into any protein. So it's not a very clean way of doing things, because But more modern studies have specifically knocked out the extra production of one or two of these target proteins, and then you get deficits in both LTP, but more importantly in real memory of real animals learning things. And as you guys were already alluding, this kind of connects back to the fish work. So the fish people were working at a time that, much before the LTP experiments, and they were the first to observe that. So there's no LTP here because this is a fish learning things. There's no slice and LTP, but the fish people, as you guys made this connection, found that the fish could learn the task initially with protein synthesis blocked. But then it would not remember the task tomorrow. So the fish was able to have early, immediate learning without protein synthesis, and it did fine, did as well as the other fish. But then the next day, this fish with protein synthesis blocked during the training or shortly after the training didn't remember how to do the task. So there's a very cool parallel between this fish experiment and what happened in the early late phase LTP experiments many decades earlier. And also, after the fish learned the task, if you wait like 3 hours and then give the protein synthesis blocker, also nothing happens because whatever proteins had to be synthesized to make the new connections had already happened in the first few hours right after the learning experience. So this is kind of a direct consequence of this fish business, but in another animal, which is like a rat or a rabbit, and in a completely different experimental setup, which is brain slice patch clamping. And this is ideally what you would want to see from scientific development, is that there's some connection to previous findings. You can replicate similar concepts across different experiments, different species, different types of measurements. Yeah.
So I'm getting a little confused in meshing these because before you said if you inhibit protein synthesis within the first 90 minutes, it doesn't affect long-term potentiation, or it doesn't affect the long-term memory. But with the goldfish experiment, it was the opposite, where if you give the protein synthesis between like immediately after and 90 minutes after the learning, then you do block.
But, sorry, which one did?
A few slides ago, there's no effect of the inhibitor in the first like 60 to 90 minutes, but there is after. But if
Yes.
you give the inhibitor...
Yeah, but that's why the goldfish is able to acquire the task with the protein blocker, synthesis blocker, while learning. But then it's kind of the transferring that into the long term is what doesn't happen.
Okay.
But here too, so this is similar to having the fish train with synthesis blocker, which is the fish is able to get the initial learning, but then doesn't remember it in the long term. And here you are able to get the initial LTP with blocking of protein synthesis, but not the longer lasting time. But so that concept is the same, but there is some kind of difference in the time scale because it's like a mouse brain in a petri dish as a fish in the water, but it's not super different because the effect starts appearing like after night, like the initial non-protein synthesis dependent thing lasts something like less than 100 minutes. Yeah.
I was wondering how we could relate this to the theory that the long-term memory would transfer to the cortex. Would that be like more dendrites forming with like chemical connections to the cortex?
Oh yeah, that's also good. So we're not getting into that, but it's also a very important process, which is that there's two things happening when the memory becomes long-term. In the case of declarative memory, like the first is that in the period of more than a few hours within the hippocampus to maintain that memory, you need local protein synthesis in the hippocampus. And then, otherwise you don't even know it the next day. But In addition to that, like you remembered, slowly that memory is getting copied into the cortex of the course of many days, and that involves changes in rearrangements in the cortex. So you get new little connections forming and unforming in cortex. So it requires more kind of slower, longer, lasting over many days, slowly happening new connections in cortex, which also would require protein synthesis, yes. Okay, so the mechanisms that could underlie LTP involve a very wide array of things. There's phosphorylation of AMPA receptors, insertion of new AMPA receptors, decreased failure rate, increased size of the response, activation of the silent synapses, and all of those are just for the early to medium phase of LTP. And for the late phase of LTP, we need to activate transcription of these new genes, which will be the raw material used to make new dendritic spines and contacts, and that requires protein synthesis. Okay, so we could either give a little break now or not do it and end sooner. So this would just be like a background of the paper reading for this week. So people want like a 5-minute break, or do you want to end the class sooner? So who wants the break? Or who wants to end sooner? Okay, so let's just go no break and end sooner. Right. So in this paper, they're trying to engineer long-term potentiation or depression, see if that was able to create kind of fear conditioning in a mouse. So the behavior is shown here. So you have the mouse in a box. Then the mouse hears a tone that's paired with a shock. And then if you put it in another box, then the mouse kind of freezes to the tone because it's afraid of this association. So it thinks the tone is very scary. And that's fear conditioning. And typically it's measured by freezing, because when the tone plays, the mouse is very scared and freezes. But in this paper, the response is measured by not increasing freezing directly, but by a suppression of lever presses to get food. So in this paper, what was happening is the mouse pressed a lever to get them food, and then the tone plays, it presses less because I'm scared, because I'm freezing. And that's the measurement they use for the conditioned fear response, which is less lever pressing, not more freezing. But the idea is the same. Okay, so what they roughly knew about fear conditioning circuitry at that time is shown here. So they know that if you just play a tone to the mouse that has no experience, it doesn't have any fear responses to the tone, because tones are not dangerous. But then if you pair the tone to a shock, now the same tone makes the mouse become freezing and scared and increases in heart rate and all that stuff. So that means that after learning, the tone is able to elicit this freezing behavior that it couldn't do before. So the behavior was known, and then to understand what was happening, there was a group at NYU, New York University, where Professor Joe Ladue and his group figured out which were the pathways that were involved in this. So they knew it's the auditory tone, so it has something to do with the auditory pathway. Then they very carefully lesion each relay of the auditory pathway until figuring out which one was blocking the fear conditioning. And what they found is that the most important site of plasticity was the lateral amygdala. Okay, so the lateral amygdala is this region that receives a convergent input of both foot shock information from the somatosensory cortex and also auditory information from the auditory thalamus and auditory cortex. So then there's this cell in the LA that's listening to both shock input and auditory input. And if those get paired, then the cell is strengthening the input from the auditory cortex. So the shock is already very strong, and you don't have to learn that the shock is bad. So the somatosensory pathway is already strong enough to activate the lateral amygdala cell because the rat already reacts to the shock. There's no learning. But when the shock is paired to the auditory tone, then you get that situation where we had the associativity LTP diagram where we had one strong input and one weak input, and the weak input gets strengthened if it happens simultaneously to the strong input. And that's kind of what's happening here. You have this strong shock input, you have this weak auditory input at the same time. The weak auditory input gets strengthened, and then after learning, the weak auditory input, which is this green synapse, is strong enough to drive the lateral amygdala to increase activity of its downstream targets, which are the central amygdala and the periaqueductal gray and hypothalamus. And then if the amygdala outputs are activated, these fear responses happen, such as freezing, more heart rate, and panic and escape and all those kinds of things. Okay, so any questions about how this works here?
I was just going to say that this makes such theoretical framework in the lecture make a lot more sense when you apply this example.
Okay. Yeah, this example is cool because it's not something specific to this strange auditory fear learning in a mouse, but first, this was the first associative learning of any sort in any species where people figured out the plasticity mechanism in some nucleus that was responsible for that specific learning. And then, if you impede flow of information, any of these pathways, it impairs the association of shock and tone. And there's also some people that have this problem. There's some woman that has lesions in the amygdala, and she cannot be fear conditioned. And if you lesion the rat, then the rat also cannot be fear conditioned. And this is not some small effect. It's essentially you get zero fear conditioning if you don't have amygdala. So it's not it modulates the fear conditioning a little. No, this is an enormous effect. It's like 100% of the freezings essentially requires this pathway. Okay, and then if the amygdala is activated by the auditory input after it's strengthened, it activates these downstream target regions in the midbrain and hypothalamus that cause like freezing behaviors and stress hormones being released and that whole thing. And that's kind of what was theoretically predicted by the Habs diagram here. So here we have, instead of this black cell, that's kind of the auditory input cell. The auditory input cell does not produce output before learning. If you pair the auditory input with this strong shock input during the learning, then you strengthen this input from B, and then B is sufficient to drive the output of the middle cell, creating fear conditioning in this case. So this was a cool example of a theoretical diagram in a book with someone thinking about what might happen, and then it turned out to really map onto real circuits in real animals learning real things that are important for survival. Because this is not some trivial thing. This is a matter of life and death, like understanding what predicts some dangerous outcome like a shock or an attack or things like that. So if this pathway is not working on you, it's like a matter of life and death. Like you wouldn't learn that touching the stove is bad or that this sound is dangerous, etc. And what they did specifically in the paper is they manipulated this auditory thalamic input to the lateral amygdala. So here we have a coronal section of the mouse brain, which means section of the brain in this direction. And then the amygdala appears here at this triangular shaped structure. And it has some subnuclei, but for our paper, the most important is the lateral amygdala, which sits at the upper tip of the triangle. And it was known, anatomically found out, that the lateral amygdala receives both of this shock somatosensory pathway and this auditory pathway going into the same cells. And it was found out that, all these pathways that we're talking about with the NMDA receptor and the CAM kinase and all those things, those are all activated in the lateral amygdala during auditory fear learning. So everything that we saw in the LTP biochemistry, like any of those pathways, if you block those in the lateral amygdala, it blocks auditory fear learning. And that's, yep.
So everything we discussed kind of only works for excitatory synapses, right? We don't really talk about inhibitory ones would get strengthened. So are all of these also excitatory?
Okay, inhibitory synapses can get strengthened as well. And there are regions that are entirely inhibitory.
But how would everything we've learned, like the mechanisms, don't they rely on you like stimulating the postsynaptic neuron? So how would the inhibitory ones?
Yeah, that's a good question. So first, the excitatory projections are plasticity is way more studied than the inhibitory. But we do know that they happen in inhibitory projections also, but they typically involve more complicated circuits that have like disinhibition and things like that. But it does happen in inhibitory synapses as well. But for the reasons you said, it's kind of, we also are, it's easier for us to intuitively think of how the excitatory synapses get strengthened rather than how the disinhibition increases, you know. And these plasticity things were done mostly in hippocampus and cortex, which are more excitatory regions, but the same pathways have been seen in plasticity in regions that are predominantly inhibitory as well. Okay, and the last piece of background that was known at that time, so they knew before doing this paper that lateral amygdala was important for learning this procedure, and then they also knew that if you do fear conditioning in a rat, then and you stimulate the thalamic input in a slice, then you get LTP types of phenomena happening. So this was, you get a rat and do patch clamping by stimulating electrically the auditory thalamus input and recording the response in the lateral amygdala cell. So you do that in either naive or fear-conditioned rat. And then first you see that the response in the fear-conditioned rat is bigger. So stimulation of the same auditory input creates a bigger response in the fear-conditioned rat. And that's shown there. See the first panel B, or say composite. The naive mouse, naive control has a smaller response than fear-conditioned control. So it's like that pathway is stronger in the fear-conditioned mouse than in the naive mouse. And then that component is also stronger in the AMPA part of the response. So if you do that experiment again while blocking an NMDA receptor, so you just see the AMPA component of the response. The AMPA component response is bigger after fear conditioning, showing that there is kind of more AMPA receptors or more phosphorylated AMPA, etc., after fear conditioning. And another measurement to show that the pathway was stronger after fear conditioning is that if you stimulate that pathway electrically, the input, you get some amount of action potentials. But if you do that same thing in the fear conditioned mouse and stimulate the auditory input, you get more action potentials in the amygdala. So the amygdala is kind of listening to the auditory input more attentively, and it's a stronger synapse after fear conditioning happens. So this is what was known at that time before this paper was done. They knew plasticity in the amygdala is necessary for fear conditioning. They knew the amygdala cells are receiving some convergent information of auditory and pain fibers. And they also knew that the auditory input gets strengthened after behavioral fear conditioning. So here what they then do in this paper is they don't use an auditory tone, but instead they use artificial stimulation of the auditory pathway paired with shock, and that causes these same behaviors, and then it also causes the same kind of plasticity mechanism in the amygdala. So what the paper shows is that kind of creating LTP in this auditory pathway to amygdala is sufficient to create this fear conditioned response, even in the absence of a real tone that was paired to the shock. Okay, so that's what we had for today. So any last questions about this? Okay, well, thanks everyone. So see you next week.